video class, let's continue to study eukaryotic cells and microorganisms. Last time we have learned about the eukaryotic cell structure. In this lecture, we are going to learn about fungi. The term fungus describes eukaryotic organisms that are spore-bearing, have absorptive nutrition, lack chlorophyll, and reproduce both sexually and asexually. Scientists who study fungi are mycologists, and the scientific discipline devoted to fungi is called mycology. The study of fungal toxins is called mycotoxicology, and the diseases caused by fungi in animals are known as mycosis. Fungi consist of an enormous group of organisms. About 90,000 fungal species have been described, although the true number of species to be about 1.5 million. Unlike protists, fungi are primarily terrestrial organisms. They have a global distribution from polar to tropical regions. Fungi are important in the food chain because they decompose dead plant matter, thereby recycling vital elements. The vast majority of vascular plant roots form important associations with fungi called mycorrhizae, which help their roots absorb minerals and water from the soil. Fungi, especially yeast, are essential to many industrial processes involving fermentation. Examples include the making of bread, wine, beer, cheese, and soy sauce. Fungi are also important in the commercial production of many organic acids and certain drugs, and in the manufacture of many antibiotics and the immunosuppressive drugs. Fungi are important research tools in study of fundamental biological processes. Many fungi are pathogenic, with over 5,000 species known to attack economically valuable crops and many other plants. Next, let's move on to the structure of fungus. The body or vegetative structure of a fungus is called a cellus. It varies in complexity and size. Single-cell microscopic fungi are referred to as yeasts, while multicellular masses are called molds. Fungi also include macroscopic puffballs and mushrooms. Like most bacteria, fungi possess cell walls. However, fungal cell walls are usually made of chitin. Instead of chitin, some fungal cell walls are composed of other polysaccharides such as mannose, galactosands, or cellulose. A yeast is a unicellular fungus with a single nucleus that reproduces either asexually by budding and transverse division or sexually through spore formation. Each bud that separates can grow into a new cell, and some group together to form colonies. Generally, yeast cells are larger than bacteria and are commonly spherical to egg-shaped. They lack flagella and cilia, but have most other eukaryotic organelles. The cellus of a mold consists of long, branched, thread-like filaments of cells called hyphae that form a tangled mass called a mycelium. In most molds, the hyphae contain cross walls called septa, which divide them into uninucleate cell-like units. These hyphae are called septate hyphae as shown in Figure A. In a few classes of fungi, the hyphae contain no septa and appear as long, continuous cells with many nuclei. These are called synercetic hyphae, as shown in Figure B. Even in fungi with septate hyphae, there are usually multiple pores or single pore in the septa that makes the cytoplasm of adjacent cells continuous. These fungi are actually synercetic organisms too. The portion of a hyphae that abtains nutrients is called the vegetative hyphae. The portion concerned with reproduction is the reproductive or aerial hyphae, so named because it projects above the surface of the medium on which the fungus is growing. Aerial hyphae often bear reproductive spores. Reproduction in fungi can be either asexual or sexual. Asexual reproduction is accomplished in several ways. First, parent cell undergoes mitosis and divides into two daughter cells by a central constriction and formation of a new cell wall. Second, mitosis in vegetative cells may be concurrent with budding to produce a daughter cell. This is very common in yeast. The formation of asexual spores often accompanies asexual reproduction and is usually used as a means of dispersal. There are many types of asexual spores, each with its own name. Atheroconidia are formed when hyphae fragments through splitting of cell wall or septum. Sporangial spores develop within a sac called a sporangia and a hyphal tip. Conidia spores are spores that are not enclosed in a sac but produced at the tips or sides of the hyphae. Blastospores are produced from a vegetative mother cell bonding. Sexual reproduction in fungi involves the fusion of compatible nuclei. Homocellic fungal species are self-fertilizing and produce sexually compatible gametes on the same mycelia. Heterocellic species require outcrossing between different but sexually compatible mycelia, called mating type. Depending on species, sexual fission may occur between haploid gametes, gamete-producing bodies called gametophyte or hyphae. Sometimes both the cytoplasm and the haploid nucleus fuse immediately to produce the diploid zygote, as seen in higher eukaryotes. Usually, however, there is a delay between cytoplasmic and nuclear fusion. This produces a dikaryotic state in which cells contain two separate haploid nuclei, one from its parents. After a period of a dieukaryotic existence, the two nuclei fuse and undergo meiosis to yield haploid spores. A drawing of the generalized life cycle for fungi showing the alternation of haploid and diploid stages. Some fungal species do not pass through the dikaryotic stage indicated in this drawing. The asexual stage is used to produce spores that aid in the dissemination of the species. The sexual stage involves the formation of spores that survive adverse environmental conditions. Fungal spores, both asexual and sexual, are important for several reasons. They enable fungi to survive environmental stresses such as desiccation, nutrient limitation, and extreme temperatures. Although they are not as stress resistant as bacterial endospores, they aid in fungal dissemination, which helps explain their wide distribution. Because spores are often small and light, they can remain suspended in air for long periods and are often spread by adhering to bodies of insects and other animals. The bright colors and fluffy textures of many moths often are due to their aerial hyphae and spores. Finally, the size, shape, color, and the number of spores are useful in identification of fungal species. Okay, that's all for today's lesson.